primary structure of a protein is a linked series of amino acids with a unique sequence. Thousands of amino acids can be linked to create a polypeptide chain. Each chain has an amino acid end and a carboxyl end. The precise primary structure for a protein is determined by inherited genetic information and not by random linking of amino acids.
Most proteins have segments on their polypeptide chains, coiled or folded in patterns that contribute to the protein's overall shape. This results in hydrogen bonds in the polypeptide backbone, which form between oxygen and hydrogen atoms. The alpha helix and the beta pleated sheets are the two main forms of the secondary structure.
The tertiary structure is the overall shape of a protein molecule due to interactions of amino acid side chains, including hydrophobic interactions, ionic bonds, hydrogen bonds, and disulfide bridges. The tertiary structure of a protein is essentially a picture of what the shape of the entire protein actually looks like.
Quaternary structure is the arrangement of more than one protein molecule in a multi-subunit complex. The bonds involved in holding these separate chains together can be van der Waals bonds, hydrogen bonds, ionic bonds, or at times, covalent bonds.
The function of transport proteins is to carry important materials to the cells and transport substances. Channel proteins allow the transport of specific substances across a cell membrane. Carrier proteins alternate between two shapes while moving a solute across a membrane during the change, just like in the picture. Hemoglobin is another example of a transport protein where it carries oxygen to body tissues from the lungs. The function of storage proteins is to store amino acids. For example, iron that is not immediately needed is stored as ferritin. Ferritin is a storage protein that regulates and guards against the adverse effects of excessive iron in your body.
Structural proteins provide support and protection for the cells in the body. Keratin, for example, is the protein of hair, horns, and feathers, and other skin appendages. Motor proteins are responsible for the utilization of cilia and flagella. Without motor proteins, certain cells like sperm cells would not be able to move or travel.
Defense proteins protect against diseases. They stop harmful pathogens from attaching themselves to healthy cells. During immune system response, antibodies attach pathogens by binding to its surface proteins as pathogens attempt to anchor to a healthy cell. By doing this, the antibodies completely block the pathogens from attaching to the healthy cell.
Hormones are messenger proteins, which help coordinate certain bodily activities. The word hormone comes from the Greek for to arouse activity. The protein hormones are first messengers secreted from the endocrine glands. Think of it as a letter with an order inside of it. There are 4 types of second messenger systems that are recognized by cells. Not only do multiple hormones utilize the same secondary messengers, but a single hormone can activate more than one secondary messenger system. In this example, the hormone activates a cyclic adenosine monophosphate system, or CAMP. This is called signal transduction, which in this case will activate the secondary effector protein kinase A.
Enzymatic proteins are proteins that speed up chemical reactions in the body. There are many different kinds of enzymes, like pieces to a puzzle, and each one helps different chemical reactions occur. Lactase, for example, is an enzyme that acts as a catalyst in the breakup of lactose into glucose and galactose, like in milk.
Proteins are created for the purpose of performing a specific biological function. Misfolded proteins that are retained in the endoplasmic reticulum are unable to perform their function, disrupting the normal biological activity of the cell.
Protein aggregation is a prominent feature of many neurodegenerative diseases, such as Alzheimer's, Huntington's, and Parkinson's disease. These diseases are sometimes called protein misfolding diseases.